Our next speaker is Steve Lindley. Steve studies how populations of aquatic organisms are affected by physical processes with a focus on anadromous fisheries. The ultimate goal of his work is development of quantitative models that can be used by resource managers to understand how populations may respond to changes in the environment. He is the director of the Fish Ecology Division of National Marine Fisheries Southwest Fisheries Science Center in Santa Cruz, and is a research associate of UCSC's Institute of Marine Science. Steve?
Thanks. So I'm obviously not Sean Hayes or Cyril Michel, which is a little bit unfortunate for this talk. They're the guys that have really done a lot of this work all the way from the all the way from the planning to the field work and the analysis. I was involved in the design of these studies and have been out occasionally with them, so I know hopefully enough to get through this presentation, which is this one. Okay. So I want to cover essentially three different studies in the brief amount of time I have here today. One is a survival of Chinook salmon smolts that are released in the upper Sacramento River, and their survival all the way through to the coastal ocean outside of the Golden Gate. So these are the other study that I want to cover is some investigations of mortality around water diversions that are analogous to what's being proposed as part of the Bay-Delta Conservation Plan. And this is just showing the map of that. And then finally, I'll cover a little bit about the ocean salmon research that the laboratory does for the past 15 years or so. And these maps just show the kind of spatial scale of the studies. So, for the basin scale acoustic telemetry studies, these began in 2007 with late fall run Chinook salmon using VEMCO acoustic tags. And we've had 5 years of information from those studies. And as of last year, we began working on other runs of Chinook salmon, the hatchery fall, winter run Chinook, and spring, including some wild spring Chinook salmon using another technology called JSATs, which allows tagging of much smaller fish. And some of the basic questions were to document patterns of survival in the river. I think at the beginning of the study, everyone knew that survival in the Delta was a big problem for salmon, and probably in the upper river and the bay, things were better. I think we found a lot of surprises, like it tends to happen when you actually go out and do the study. So this just shows some of the different habitats ranging from the upper basin here, which is a relatively, there's a laser on here somewhere. There, yeah. The upper river, which is a beautiful environment for natural channels, looks somewhat pristine when you're floating down a boat, until you get into the lower river, which is a much more channelized and modified environment, and then to the Delta, where you've seen a lot about that today. And then out into the bay and the ocean, which once again looks more like a hospitable and natural environment. And this shows the familiar salmon life cycle. I think everybody here knows about the migrations and the development of these animals. So looking over the 5 years of the late fall run Chinook salmon at survival rates that have been normalized to 10-kilometer reaches, and they're arranged from the wrong button. Uh-oh. That wasn't so bad. The upper river is in white here, and the lower river then, which is basically below Red Bluff, where the channel morphology changes and it becomes more modified channel banks, in the dashed, the Delta in the broad hatching, and the bay and the closer hatching. So we see fairly consistent patterns over space and time within these broad geomorphic areas with moderately, well, it appears moderately high survival, but keep in mind these are per 10 kilometers. So these are actually significant levels of mortality in the upper river that are going on. The lower river, quite counterintuitively, had quite a bit higher survival, including some reaches where salmon almost never suffer mortality, apparently, even though these are rip-wrapped channels. The Delta has moderate mortality, as we would have expected, not too far out of line with what we see in the upper river. And the San Francisco Bay has substantially higher levels of mortality, which was somewhat of a surprise. Over the 5 years of the study, there were 4 years which were pretty much dry, and the first 4 years, and the final year which was wet. And we do see consistent patterns, more or less. Now this survival here is cumulative survival, and the reaches are aligned according to the actual distance along the river. So you get a feeling for the patterns of accumulating mortality over time. So we see in the dry years a fairly rapid die-off of the fish as they're moving down in that upper river. They hit the lower river. There's very good survival through here. And then through the Delta, the numbers drop again, finally reaching fairly low levels of survival to the ocean of 5 to 7%. During 2011, which was a wet year, survival through this upper river reach was quite good. There aren't data points in the middle because the detection probabilities were poor. I'm not quite sure why that is. If it was due to the rapid movement of the fish through the reaches where the hydrophones were, or perhaps turbidity or other things, noise is masking the detection of the tags. But regardless, a lot of fish did make it out of the upper river and then subsequently suffered pretty decent levels of mortality. And then in the bay, they suffered very similar levels of mortality as they had suffered during the dry years. So, but they did survive about threefold higher. Okay, moving on. This is some information about the question of whether travel time or distance is important. And they did move out a lot faster in 2011, the wet year, than they did, roughly maybe 70% as much time taking to get down the river. And that could explain part of why they survived better. I'll talk about this a little bit. I'll be back to talk about life cycle modeling and predation, and I'll have some more of a theoretical treatment of that briefly. But anyway, travel time is one aspect of survival. So this just compares the travel time and survival rates of the different runs of fish. So this is very preliminary results for fall, spring, and winter, just 1 year of data, and not huge sample sizes. We had some technological issues with the JSATS tags that were getting ironed out. But one of the kind of surprising things here was the variability in survival rates among the different runs of salmon. So there were some questions earlier about whether one fish is a very good surrogate of another. I think this, we're going to learn, is generally a kind of a dangerous thing to do. Ideally, we would be tagging the animals that we're really interested in if we can catch them or get permits to do it. And we see that the late fall had this 3 to 16% survival and take about 15 to 30 days to get out. The fall run Chinook salmon, not surprisingly to me, had lower survival. These are much, much smaller fish. So we would expect small fish to survive perhaps less well, but they move out incredibly fast. And you might think that larger fish, being better swimmers, would move out more quickly, but actually these fall run Chinook salmon move out very quickly. The spring Chinook salmon released from the Feather River had very poor survival. And also there's differences in the spatial pattern. They really suffered very heavy losses within the Feather River itself and did better once they got out of there. And the winter run Chinook salmon, which is more of an intermediate-sized fish migrating in the springtime, had not very good survival either, and took quite a long time to move out of the system. And for some reasons that we don't understand yet, after release, they moved downstream for a while fairly quickly, like the late fall run Chinook salmon do, but then they take up residence in the sort of middle Sacramento River for some period of a month or more before they then resume their migration. In the meantime, predators are doing their seasonal things as well, and so each of these fish with their different life histories is probably exposed to different patterns of predation in the environment. Okay, by way of some context, looking at other systems, what does a 5% survival mean for a Chinook salmon population? We worked with some of our colleagues in the Columbia River and the Fraser River in Canada to look at survival rates there. The Fraser River is relatively, compared to the Sacramento River, I would say, an unmodified system. I think it suffers some land impacts from logging and whatnot. But they see survival rates sometimes quite low, but substantially higher there, perhaps not at all surprisingly. What's a bit more surprising is the Columbia River, which is a poster child for salmon systems that are being heavily impacted by dams. They see quite a bit higher survival there, sometimes quite a bit higher than even 28%. So we're seeing much, much lower levels of survival in the Sacramento River, and the question is really why, what's going on there. We don't have the dams, which were thought to be the big problem in the Columbia River with the passage problems for the hydro facilities, yet our salmon are doing far worse. So, moving on to the next study. This was motivated by the Bay-Delta Conservation Plan, which has part of its proposed design a number of large water diversions in the lower Sacramento River, which would include state-of-the-art fish screen facilities. And the idea is that they'll have no direct impact on salmon. The screens are such that the fish will not be impinged on them or entrained. Yet these structures may attract predators, and the filtering action of the screen itself may concentrate prey in local areas, and you can imagine this could create a predation hotspot. And if that were to be a large effect, a lot of the benefit of moving the pumps into the lower river with these state-of-the-art facilities would be negated. So we did 3 years of work on this, looking at some analogous structures, one near the city of Sacramento's intake facility near the confluence with the American River, and then just a bit downstream, the Freeport water diversion. This is a picture of the Sacramento diversion, which is kind of an island out in the river. The Freeport diversion is on the bank. And let's see, some more pictures of where we were at. I wanted to show you this, kind of a conceptual model of how water diversions might interact with predation to create survival issues for salmon. And one has to do with the effects of the water entrainment, which can lead to this smolt aggregation through this filtering thing, and then the predators responding to this prey aggregations, but also the physical structure itself leading to aggregations of predators, then these things interacting in these local environments leading to the predation and subsequent mortality. And we have different aspects of the study designed to look at each facet of this. And I'll cover 3 of them, I think, today. One is just looking at the density of predators around these sites, and you saw some pictures. I think Jacob showed some Didson camera results, and we used very similar techniques to scan around the facilities and adjacent areas near and far away from the facility to count predators. And we did this day and night, crepuscular periods in the middle of the night where we could. Turns out around the Freeport, or actually the Sacramento diversion, there's a big angling community there that weren't great fans of this work, and they really fish all times of the day and night. It led to some interesting logistics and interactions with the public. Freeport's doesn't have those problems for some reason. So this is just a picture again of what you get out of a Didson. It's not that compelling on a still image. The videos are actually remarkably clear in the way of an ultrasound, if you've seen those when they look at babies and whatnot. You can see the motion, and you can tell really that there are fish swimming, and this just shows the image. Often the echo shadow is what's really visible. You can get a more or less reasonable estimate of the size of the fish that you're seeing and classify them over a certain size as being possible predators, with the caveat that Jacob mentioned that there are carp and shad, which may be that big but don't probably eat salmon. So this shows a bit about how the transects were done around the Freeport diversion. This is where we would take the boat along here and scan along these lines. Expecting most of the predators to be along the bank, so concentrating efforts there, but doing these cross-channel transects to look at the more open water areas. And this is the City of Sacramento diversion with intensive monitoring right around it and the similar transects. And the little dots here are predator-sized fish that were observed there are a lot of fish in the river, which may explain why, this is the park here where the recreational anglers, or I guess they're subsistence anglers really, are fishing. I guess they figured something out some years ago. And Cyril divided this up for his analysis into zones of the near bank and the middle of the channel zone to look at some aggregations overall. And we see some moderate predator densities. Generally, the banks have higher than in the middle of the channel. And for the case of Freeport, there do appear to be somewhat higher densities of predators near this structure, where in the Sacramento River, they don't really hang out around that structure as much as they do. And that's the one that's out on this sort of artificial island. Similar levels of predator densities at both sites. So another thing that we did was try to look at the diet of predators using some tethering experiments that I think I'll cover if I remember what's in this presentation correctly, and also hook and line angling, which is a popular volunteer job around the laboratory to go out and do this. The tethers are basically little floats that are anchored on the bottom and have a hook timer and a leader coming off with a live salmon smolt on a small hook. And those were deployed for an hour. And at the end of the hour, you pick them up and see if there's a predator on the hook or if the hook has been snapped off. And then that indicates there was a predation event or whether the smolt is still there. So overall, they caught a large number, or a fair number of fish anyway, striped bass being the most abundant thing here, but also some pike minnow and centrarchids. And I think kind of a typical thing, there's always a proportion that have empty stomachs, and then something with unidentified thing. And we actually did detect some fish that had salmon in their bellies. At one point I did want to make that Sean put in this presentation, but I didn't think I'd have time to get to, so I took it out. He did some bioenergetic calculations and considerations of the abundances of striped bass, salmon, and whatnot. And even if the striped bass were to eat all the salmon, you would rarely find them in the bellies because there's so many other prey items out there. They're a minor element of the prey, but that's not an argument to say that striped bass can't be a problem for salmon. It just makes it very tough to study. Okay, we've also been interested in whether the predators at these sites hang around there all the time or whether they're coming and going. They're just transients that take up temporary residence there. So we've been tagging the predators that have been caught with the familiar by now acoustic tags. And they've found that the striped bass are coming and, that's probably the next slide, are coming and going fairly rapidly. And the pike minnow tend to be more resident at these sites. So it's a bit of a problem that if you wanted to try to get rid of the predators near these sites through fishing them or whatnot, the striped bass will tend to just keep coming back, I think. Okay, this is a little bit more about the tethering results in detail. This is a schematic of the apparatus here with the hook timer. These were soaked for an hour at different, these are the stations here, so these are laid out mostly along the bank and around the structures, but some also in the middle channel. And a number, I guess 6 different tethering events were done in crepuscular periods as well as the middle of the day and the middle of the night at these different sites. And these are measures of relative predation rate, the bigger the circle being the more probability that the tether recorded a predation event. And there's not a super obvious pattern that we can really detect from this yet. Really, in spite of the fairly high abundances of predators at this facility, oddly enough, the smolts on the tethers didn't suffer very high loss rates there, just generally along the banks. The absolute magnitudes of these range up to about 50% for a 1 hour set. So that's a pretty high risk of being eaten, undoubtedly a lot higher than a free swimming smolt. So these are really only useful for relative comparisons The half-life of wild salmon has to be a lot more than an hour, or we would never see them. So there are the results here. Okay, this is some information about the seasonal variation in predators and predation. And we do see a pattern of increasing predator abundance in May. This is coincidentally the peak of the salmon smolt migration. That's a little bit alarming. The predation rates itself, as reported by the tethering experiments, don't really vary in any kind of super clear pattern. And the seasonal pattern was fairly consistent. So this was, Jacob already covered this essentially. This is the idea that we're going to go out working with DWR and other collaborators and remove predators from some significant areas of the delta and use the same kind of methods that I just talked about around these water diversions to see whether you can really get rid of predators, how quickly they would come back, and whether you can get a benefit for salmon survival. I'm most excited about this study for about what we'll learn about the predators that we catch during the removal, about their habitat associations, their diets, their identities, really, for that matter. Okay, I'll skip through this and finish up here with the ocean trawl surveys that were begun by Bruce McFarland at our laboratory. He's now retired, and Sean has taken over this work. And this sort of shows where we're doing this and when. And the basic idea is to follow the cohorts of salmon as they move from the bay into the coastal ocean. So in the springtime, they sample in San Francisco Bay, just inside the Golden Gate. And then in the summertime, they trawl using NOAA vessels or charter vessels in this area where the young salmon are thought to reside before they eventually move north and south. And to repeat that ocean sampling again in the fall. And in principle, this could tell you something about predation in the ocean by if you can form regional abundance estimates and seeing how those change over time. Whether the sampling's going to ever be up to that, I'm not so sure. The catches are pretty patchy, not surprisingly, and salmon aren't the most abundant thing in the big ocean once you get out there. But what you can do is learn a lot about the salmon that you do catch. And historically, Bruce's focus on this was on their energetic composition, their lipid content, their size, and things like that. He had a kind of a physiological perspective. The other thing is that we've recovered the otoliths from these. And I think we've learning some pretty fascinating things about that. So most of you know about fish otoliths. They lay down daily growth increments like a tree lays down annual growth increments, and you can count these up to get the age of the fish when it died, when you caught it in your net. And the increment spacing tells you something about the growth rate and the size at age. You can back calculate their size at age. And the idea of the study that's been just coming out in Marine Ecology Progress series, we'll have the citation in a couple slides, is that you can get an idea about size selective mortality, which is probably driven by predation, by looking at the distribution of something, say, at the size at age or the growth rate at some age of a population at its entrance to the ocean, and then come back later for the survival, for the survivors. And if there's been a shift in this distribution, it would indicate size selective processes going on in whatever your measure is. And essentially what they found is that during years when we know survival was bad based on Côte tag recoveries and what we think are poor ocean conditions for salmon, that small salmon entering the ocean do very poorly. And during years of good ocean conditions, they all do relatively the same and probably better, presumably. They must be doing better. So we see this shift in the size distribution over the season during these poor years. And this is the citation for Lindsay's paper, which is in page proof right now, so if you want to see a copy of this, I think I can get that for you. And I think I'll end with that. Thanks. Any questions? Thanks. Do we have a minute, or is this?
We have just a minute. We're running a little
Okay.
behind, but if you have one question.
Yeah, question about the acoustic tag work, and it may apply to some of the other presentations as well that used acoustic tags. I'm wondering if you've got an idea as to the precocious maturation rates of the salmon that were used. So in the Columbia, we're finding really high percentages of the fish that are sampled, both in, well, particularly in hatchery populations and some in natural populations that are maturing in fresh water. So some
As yearlings.
Yeah, and they will, and sometimes as subs. So sometimes they will residualize in the area near the release. Sometimes they will migrate all the way down to the estuary and then reascend the river. So I'm just wondering if some of the results, you know, you had mentioned some of the fish went down and then they kind of held up and had some behavior that may not have been fully expected. I'm wondering if some of these might be explained by precocious maturation of these fish.
Yeah, that's a great question. I don't think so. Not for Chinook. It's definitely an issue for the steelhead. We actually, when we first got the results the first year, we were surprised. Where did they go? We didn't expect that kind of high mortality. So we actually floated the river with hydrophones, thinking maybe they've just taken up residency, and we did not find any. And we have seen them come back as 2-year-olds. The tags have lasted that long. So we've seen some fish ascend, but you know they're jacks. They're not these yearling precocious males. So I don't think that's going on. Someone will correct me if they've seen otherwise, but I don't think it's an issue.
Just for reference, in the Columbia, we've seen precocious maturation rates amongst males of over 70%, and pretty much everywhere that we look, we find them. So just something for reference.
Thanks.
I had a question about the, sorry.
I'm trying to keep you on track.
I know, I'll ask this one quickly.
You can make it up in my next talk.
The sort of relatively low overall survival you're seeing in relation to other systems, and there's potentially different tag effects depending on the type of tag. So, and some with the lower frequency tags possibly hearing by pinnipeds, and some of the higher frequency tags, unclear mechanisms, but a really clear difference in relative survival compared to acoustic tags and pit tag fish on the Columbia, upwards of 20+%. So do you think any of your survival estimates are possibly influenced by any of these tag effects?
I don't think, we don't have the pinniped problem in the river. That there aren't those kind of sites where they can do that. So I don't think that's going on, but there could be tagging effects. We don't have the pit tag double studies, you know, that we don't have ways of detecting the pit tag fish to be able to do that. It would be a really great thing to do, because these tags still are, they're pretty substantial. We do hold them in tanks, and they survive fine, and we've done swimming performance tests, and they swim fine, but it's, the steelhead have had problems of tag rejection. We haven't seen that with the Chinook. So it's an issue that's unresolved. Thanks.